Hello, everyone. Welcome to the Events, Memories, and Language oral session. I'm stepping in as chair. I'm Monica Rosenberg, an assistant professor of psychology at the University of Chicago. And we're going to go ahead and get started. I'm excited to introduce our first speaker, Loic Labache. Because I am filling in as chair, I do not have the ability in the Q&A part of the app to remove questions at the end of each talk. So when you ask a question for each talk, please start your question with the number of the talk. So when you ask a question in the app, please start your question with 1, and we'll also welcome questions at the microphone after the talk as well. Okay, well, thank you. Take it away, Loic.
Hey, hello everyone. So my name is Loic Labache, and I am a postdoctoral researcher at Yale University in the Department of Psychology. And today I will talk to you about the relationship between language lateralization and global brain architecture. So just to get everyone on the same page, we will define hemispheric specialization like a relationship, an interdependent relationship between a set of brain structure in one hemisphere and a given cognitive function. So as you all know, hemispheric specialization is like a fundamental principle that is found through the entire animal kingdom. And today we will engage these hemispheric specializations through the perspective of language lateralization. So first, we can define like a typical phenotype language organization as like a left dominant hemisphere for language. So that means if anything happens to this hemisphere, it will probably lead to aphasia, and we can find typical language organization with an equal probability either in right or left-handed people. But the problem is that we don't have a lot of information about the relationship between all the different lateralized cognitive function. So I will try to answer to this question, like what is the link between all the lateralized cognitive function. But first, to do that, I will have to define what is a typical heteromodal language network. And next, I will just try to define extensively what are like the properties of typical and atypical brain organization. So, as I said, first, and in 2019, I published a paper. Like the paper was about the language network atlas, and to do that, I use like the Billie Jean database. It's like a small database with 144 people that perform 3 different language tasks: a production, a reading, and a listening task. And to make the story short, I just perform a conjunction analysis of these 3 language tasks. So I selected the brain regions that were both activated in the left hemispheres and leftward asymmetrical in the free contrast, and I came up with this 32 supramodal brain region in the left hemisphere, and all these regions will define the language network that we will use next to characterize individuals. So, as I was saying, there is a lot of crucial information about like the organization of hemispheric specialization in atypical individuals that is missing, like Wimber said it in his paper in 2019, such for example, the regional activation pattern of atypical individual and a lot of other things. But the question that we can ask is like, why all these things are missing? So first, because there is a really low prevalence of atypical people. So knowing that typical people are people with a left hemisphere dominant for language, the atypical people are just the people that have a right hemisphere dominant for language, and they are really rare to find in the general population. And the second fact is that this atypical population has a highly variable hemispheric specialization pattern, with some people with classic right hemisphere dominance, but some people have like the 2 hemisphere dominant, or some people don't have dominant hemisphere at all. So it's kind of tricky to identify them. So what I did in this talk is like I examined the functional organization of the language network in the dominant hemisphere and its interhemispheric coordination with its homologue in the opposite hemisphere. And to do that, I take advantage of the Human Connection Project database. So I am working with 995 participants that have both performed a language comprehension task and a resting state fMRI acquisition. And I characterize their language network. So I have like 5 biomarkers for the language network. I have first the functional asymmetries induced by the language task at the network level, as you can see on the right picture. And I also have the language asymmetry, the functional asymmetry at the hub level, so the Broca and Wernicke area, for example. Broca and Wernicke. And I also have like the intra-hemispheric intrinsic connectivity strengths. I have both the strength asymmetry and the strength sum for the language network. And the last biomarker is like the interhemispheric homotopic intrinsic connectivity. So it's like how the 2 hemispheres are communicating between each other during resting state. So I just perform a hierarchical clustering on these 5 biomarkers for these 995 participants. And what I found is like these 3 clusters. As you can see in orange, it's a cluster of like really strong typical people. So these people are really like a strong leftward asymmetry during the language task. And I have another cluster of mid-typical people. They are also like left dominant, but this one has a more like moderate left asymmetry, leftward asymmetry. And finally, the group of people that are for interest today is like this cluster of blue people on the right. And these people represent approximately like 8% of the sample, and they are like the atypical people with a strong rightward asymmetry. And what is interesting is if we look more into the detail of each of these clusters, we can find the same results that I previously published with another database. So I think we can conclude for that like using this method of 5 different biomarkers for the language network is like a robust method and it's reproducible. And you also have to note that atypical people do not have a brain organization that like completely mirroring the organization of typical people, mainly because when the typical people has a leftward asymmetry during resting state for their language network, atypical people have basically a symmetrical brain organization during resting state. So it was just a little comment about it. So now that we identify this 3 groups, these 2 groups, this group of typical and this group of atypical people, we can ask if the lateralization of a specific function, so in our case the language function, is reflected across the architecture of the sensory flow integration of the entire cortex as defined by Mesulam in 1998. And to answer this question, we take advantage of the mathematical modeling proposed by Daniel Margulies in 2016. So as you may aware, we can, using like a nonlinear ACP to compute the first 3 gradients of the brain. And to do that, we can find the cortical organization proposed by Mesulam in 2000. So just for a remember, the first gradient is anchored on one end in primary area and in heteromodal association cortex on the other end. The second gradient opposes the primary visual cortex to the somatomotor auditive one. And the last gradient opposes the internal versus the external processes. So because we wanted to keep the analysis simple, what we did is computed these 3 gradients for all the participants, but we averaged the gradient value for each of the 7 networks proposed by Thomas Yeo in 2011. And what I did next is I just computed the average asymmetry for each of these networks and for each participant. So basically the left hemisphere minus the right hemisphere. And finally, I just perform a small ANCOVA to determine the impact of language phenotype on gradient asymmetry. So basically what we want to see is if the typical or atypical organization has any impact on this gradient asymmetry for each of the 7 networks. And when we are doing that, The results are on this graph on the right side. And what we can find is like for the free gradient, the association cortex is the one with the strongest typicality effect. Yeah, as you can see, like the default mode network, the control network, and the salience ventral tensional network are the ones that are like the biggest t-statistics. But the other point that is really interesting is like for the third gradient, the effect of typicality is like the opposite effect. That means that atypical participants for the third gradient are leftward lateralized, when the typical ones are rightward lateralized. I still have to work on the interpretation, but it's an interesting one. And what I did next is I tried to find if this gradient asymmetry and language phenotype was heritable or not. And to do that, I just perform a classical heritability analysis on a subsample of the HCP database. This time I have only 989 participants. And what I found is like the language lateralization phenotype, so the fact to being typical or atypical, is 13% heritable, and it's significant, so it's pretty cool. And what I also did, it's like I performed an heritability analysis directly on the gradient asymmetry. And what we can find is, once again, the heteromodal associative cortex is the most heritable, with an average of heritability at 19%. And once again, the third gradient is the one with the most, is the one that shows the most heritability, like before with the typicality effect. So from that, we can conclude that both language lateralization and gradient asymmetries are, at least in part, driven by some genetic factors. And I think what you have to remember from all these slides is like the presence of language network atypicality parallels the macroscale organization of the entire cortex, spreading from the first gradient to the third one. And that the canonical language network appears to be only one component in the broader hemispheric organization of the human brain, and that this language network reflects the specialization of distributed large-scale networks across the entire cortical sheet. And finally, that the language and the large-scale brain functional organization is highly heritable. And we have still some questions. Few questions that we can ask next is to define the exact role of the corpus callosum in the interhemispheric regulation of this language, and more globally, like the hemisphere organization and regulation. And we can also ask what are the mechanisms that are supporting the implementation of these asymmetries during the development. So thank you all for your attention, and I would like to thank not my team, but Aram Holz and Tian and Thomas Yeo that have worked on that with me. Thank you.
Thank you so much for the talk. We have questions in the app. So the first question is, what about more balanced bilateral organization, which is another type of atypical lateralization? Did any of your samples show this?
I suppose that you mean like people with both hemisphere dominant. So basically the mid typical group, I didn't go into too much detail, but they have a tendency to show, yes, like a bilateral organization. Even if in the HCP database, they still are leftward lateralized, but it's not really clear. But in my previous paper that I published in eLife using another database, I found some really symmetrical people, and these ones have a tendency to have the same organization than typical at rest. So, at least from my point of view, the ones that are really interesting are like these atypical people with a weird organization.
Thank you. And this is a question I also had, which is task data needed to distinguish between language phenotypes, or is rest data sufficient to do this?
Or do it's better to use the task or the resting state too? So actually, if you perform like a hierarchical clustering using only task data, it's not impossible, but it's more difficult to find the atypical people. So I think the best way to do it is to combine both resting state, and task activation, because I didn't show it there, but this atypical really doesn't have the same organization during resting states than the typical one. So I think it's better to take advantage, I mean, if it's possible, to both like task language activation and resting state.
Great. Well, thank you so much. Let's thank our speaker
Thank you.
again. And up next, we have Edward Silson. Also, please remember to, when you ask a question for this talk, start your question with talk number 2, so we can keep track of the questions associated with each talk.
Well, good morning. So memory recall recruits a broad array of cortical regions, ranging from the hippocampus to medial parietal cortex, highlighted here in green. Now, although historically this region has been considered as largely functionally homogeneous, our group recently identified a finer-grained organization within medial parietal cortex, with an alternating pattern of regions recruited during people recall, shown here in red, or, place recall, shown here in red, or people recall, shown here in blue. Now, this effect manifests as 2 pairs of regions, one larger and more ventral, which I've termed places 1 and people 1, and a smaller, more dorsal pair of regions, which I'm going to call places 2 and people 2. Now, the evidence for people and place representations in medial parietal cortex has subsequently been confirmed in ECOG data and additional fMRI data independent of our group. And so this convergence sort of begs the question, well, what is the underlying functional organization of medial parietal cortex? Well, the spatial adjacency of people and place representations in medial parietal cortex is strikingly reminiscent of the categorical shift, the categorical preference shift that we see in ventral visual cortex. So on the left-hand side of the screen, you can see a ventral view of the right hemisphere. And what we know from decades of research is that face representations in the fusiform face area, or FFA, shown here in blue, are always lateral to place representations in the parahippocampal place area, or PPA, shown here in red. And so this consistency in the spatial arrangement of these representations led us to propose a framework which was to ask whether or not medial parietal cortex reflects the categorical preferences along this medial to lateral axis in visual cortex. But of course, visual cortex and ventral temporal cortex in particular is more than just people and places. So for example, object-selective posterior fusiform, shown here in green at the bottom of the slide, sits typically in between face representations in the FFA and place representations in the PPA. And so if you take this into the framework I just outlined, what it would predict is that during object recall, you should not only experience or not only recruit distinct regions within medial parietal cortex, but that those regions should fall in between the place and the people regions we showed before. So this is exactly the question we asked in this experiment. So here, participants in the scanner were cued with a word stimulus to a personally familiar person, place, or object. And they simply had to recall the target from memory as vividly as possible. Okay, so no images were shown here, they were all just word cues. So this design hopefully allowed us to accomplish 2 goals. So the first was a direct replication of our previous people and place data, and the second was to establish whether we can find these distinct regions for object recall within medial parietal cortex. So what did we find? So let's just start with the replication. So at the top of the screen, I'm showing you medial views of both hemispheres with the contrast of people versus places overlaid. So place recall here is in hot colors, people recall is in cold colors, and what I hope you can be convinced of is that here in this independent data set, we've still got this alternating pattern of people and place recall. Now we can look at this in a little bit more detail by pulling out the 2 pairs of regions I spoke about before. So starting with the ventral larger pair, On the y-axis here, I'm plotting just the mean response to each category. And what we can see is that in places one is really only ever driven above baseline during the recall of specific places. And in people one, it's only really driven above baseline during the recall of specific people. We see a very similar pattern in the smaller, more dorsal pair of regions, albeit reduced in magnitude. So for us, what this constitutes is a full and independent replication of our prior work and is consistent with the ECOG and the fMRI data I mentioned before. Okay, but what about objects? So at the top of the screen, I'm showing you the same medial views as before, but now with the contrast of places versus objects. So you can clearly see we get robust place recall effects shown in blue, and objects effects, if they were there, would appear in hot colors. But what we're seeing here is a distinct lack of any object recall effects at this statistical threshold. The same lack of object recall effects is present when we compare people with objects. And so it appears here that at least within medial parietal cortex, we have no evidence for any object recall effects directly. But this lack of object recall, as well as being slightly disappointing, was also a bit surprising for us. So we wanted to look outside of medial parietal cortex to see, do we actually see anything to do with objects? So at the center of the screen, you can see lateral views of both hemispheres. And what I've overlaid in hot colors here are just the main effective category from our fMRI modeling. And what this highlighted was several regions of interest that we wanted to follow up in post-hoc analyses. And these included the posterior STS of the right hemisphere and the putative frontal eye fields bilaterally. So to look at effects within the posterior STS, we wanted to identify a region of interest using independent data. So we took advantage of a dynamic localizer for the posterior STS, which was the presentation of the significant response to dynamic faces versus static faces. So again, the y-axis here is just the mean response to all categories. What we see in the STS is that on average, all responses are negative during recall, but they nevertheless differentiate on the basis of category and are significantly less negative during the recall of people versus places and objects. To quantify effects in the frontal eye fields, again, we wanted to do this independently. So we took advantage of a retinotopic probabilistic atlas for the frontal eye fields. Again, the y-axis here is just mean response. And what we see in the FEF is that responses are really only driven during the recall of places over people and objects, which to us suggests that this might reflect different eye movement behaviors during the recall phase. Okay, but it's not all bad news for objects. We do at least see something. So again, on the lateral surface here, this is the contrast of people versus objects. And what I want to highlight is we do see a small cluster of object-related recall in the vicinity of the supramarginal gyrus, which has been implicated for a long time in representing tools and reaching behavior. Okay, so just to quickly summarize, what I've shown you here today is that we have provided an independent and complete replication of our prior people and place data, and just eyeballing these two medial surfaces, you can see how strikingly similar these data are. We found no evidence, or at least no significant evidence, for object recall effects within medial parietal cortex itself. We found some people recall effects in the posterior STS of the right hemisphere and place recall effects in the frontal eye fields bilaterally. So what do I think all this means? Well, it's a little bit disappointing for my framework of mirroring medial parietal cortex being a mirror of ventral visual cortex, but it suggests to me that perhaps the categorical memory effects in medial parietal cortex are restricted to those to do with people and places. Now, why might that be? Well, we know that faces and scenes elicit the strongest responses in ventral visual cortex, and indeed elicit the strongest contrast in responses. And so it's possible that what medial parietal cortex is doing, if it is at all reflecting ventral visual cortex, is it's reflecting only the most salient categories represented there. One possibility also is that the reduced semantic, emotional, and/or contextual associations with our objects category could underlie potentially the lack of responses in medial parietal cortex. But I will preempt any question about how vividly participants could recall these by saying there's no difference in subjective vividness across these categories. Okay, so I'd just like to thank my co-authors, the following institutions, and finally, thank you all very much for your time.
Thank you for the talk. So we have many questions in the chat. First, why did you use written representations rather than images in your task? I would imagine you saw more visual word form area activation than activation in other areas.
So we do see visual word form activation, but there's no that doesn't differentiate on the basis of the category. The reason why we used word stimuli is because we wanted to minimize any impact of the actual bottom-up visual input. We wanted this to be memory recall and mental imagery. So we wanted to avoid any swamping of the bottom-up effect that you would get if you showed actual visual stimuli.
Thank you. And how do you think this organization would look under more naturalistic conditions instead of evoking these regions with sort of static input?
Sorry, one more time?
Oh, sorry. How do you think this organization would look different, if it would look different, if you, under more naturalistic conditions?
So I don't think it would look different at all. So the ECOG paper that I referenced very quickly at the start, they actually presented real images of faces and scenes. So I know that's not real world in a sense or naturalistic, but it's more visually driven than the ones that we presented, and they still find this same repeated organization. So I don't think it would have changed the overall pattern or the overall topography of the findings that we had.
Got it, thank you. And is there any evidence from lesion studies for places to and people to areas in MPC?
So it's a really good question. Not that I'm aware of, although there are some cases where you can have Capgras syndrome, which is not only the sort of illusion that you're meeting someone who's an imposter, but you can always also have that for places as well, and that's generally considered to be caused by lesions to medial parietal cortex. The problem being is that these regions are quite close together in proximity, and lesions usually cover a large diffuse area, so it's very difficult to pinpoint exactly where that might be. But yes, there is some evidence.
And then we have another interesting question in the app, which is, why do you think there's so much variability across participants in this organization?
That's a good question. Well, one of the things that I can tell you what I think it can't be. I don't think it can be due to the subjective vividness, because that's equal across categories and across people. What it might have to do with is the fidelity of the connections between visual cortex and medial parietal cortex. So the previous paper that this is all based on, we demonstrated that there's this functional connection between face and place regions in visual cortex and the subsequent memory areas. And so if you have larger responses or more distinct responses in visual cortex, that might allow you to drive the memory regions more during recall, and it might just reflect the natural variability. One of the things I want to do in follow-up studies is look at aphantasic populations, so they have an inability to use mental imagery, and if this framework is correct, you would anticipate that connection between visual and medial parietal cortex would be significantly weaker than in normal individuals.
Great, well thank you so much. Let's
No problem.
thank our speaker again. And our next talk is from Linda Geerlings. For this talk, remember to start your question in the app with talk 3.
Hi, everyone. So today I'd like to talk to you about the neural mechanisms of event segmentation. So I want to start off with introducing this concept of event segmentation by asking you this question. How has your day been so far? So take a moment and think about this. Now, probably as you're thinking about this, you're realizing specific events that happened in your day. So maybe you thought about having breakfast this morning, maybe you walked over to the conference, went to the keynote lecture, maybe you grabbed some coffee, and now you're here. So this is really a characteristic feature of how we process this continuous stream of information that comes in through our senses, is that we chunk it into these meaningful units or events. And this has a lot of advantages for how we memorize information and also for how we process information. So first advantage is that it gives us a very compact representation of these extended periods of activity that we engage in. And second, these moments between distinct events, so the boundaries between them, are key moments where we store information from the previous event into long-term memory. But it also has advantages for how we process information at this moment. So the boundary between the previous event and the current event allows you to free up working memory that was related to the previous events and to reorient yourself to the current context. So I'm interested in studying the neural mechanisms that underlie event segmentation. The question is, how do we study this in the lab? So one tool that we can use is to use narratives. So these narratives are basically condensed representations of real-world-like events and interactions. So they have very high ecological validity, not only because they represent things in the real world, but also because we are used to engaging with narratives like movies or audiobooks or stories. And what we can do with these narratives is to ask people to indicate when, in the lab, like when does one meaningful unit begin and another one end. And people are able to do this quite reliably. So different people tend to indicate the same moments as transitions between events. So to investigate the neural mechanisms of event segmentation, Chris Baldessano and colleagues really came up with this concept of neural states, in particular brain regions. And here I just want to quickly illustrate this idea of what a neural state is in this context. So a neural state is basically a brain activity pattern that is stable for a period of time before it then transitions to a different pattern. So here we can identify these three distinct neural states in this hypothetical example. But now let's look at some real data and let's look at what these neural states might really look like. So here we see data from the medial prefrontal cortex. And so what you can see here is a time-by-time correlation matrix. So we took the voxel patterns at each time point in medial prefrontal cortex and then correlated it with all other time points. And what you can see here, I hope you can see my pointer, so you see that the voxel activity pattern here is pretty stable for a while. So if we look at correlation with the first time point, it's pretty stable, and then suddenly there's this transition, right? So there's this sudden change in the voxel activity pattern. And here we see the same thing again, so it's pretty stable for a while, and then there's this transition again. So important to realize here is that this is data not from single subjects, but this is data from people who are watching a movie, and data that's averaged across a bunch of participants. And that really allows us to see this really, really distinct transitions between these states. So what we can do here is now to apply data-driven algorithms to identify these boundaries between distinct neural states. And so Chris Baldessano and colleagues, they did this in their neuron paper, and what they showed is that there's this interesting alignment between these data-driven identified neural state boundaries and the event boundaries that people rated subjectively in an independent sample. And they also showed that there's these different durations of neural states across different levels of the cortical hierarchy. So lower levels of cortical hierarchy, like early visual areas, would show very brief neural states, whereas higher level areas like medial, like the precuneus, would show very longer lasting neural states. So based on this, they came up with this hypothesis that there should be this nested temporal hierarchy of neural states that underlies our ability to segment our experiences into these distinct events. So the idea is that each brain region that chunks experience at its own preferred time scale. And so this might correspond to things that actually change in the external environment. So for example, for a low-level auditory region, might chunk at the level of words, or a high-level auditory region at the level of sentences. And importantly, these boundaries between these neural state transitions, they're nested. So that some of these states, they actually propagate up in the hierarchy. So you see them across different cortical levels of the cortex, whereas some states don't propagate at all or propagate only partly. And those that propagate upward would also be the ones that we actually perceive as being a boundary between events if we experience things subjectively. So based on this framework, we came up with a number of hypotheses. So first of all, we would expect that there is this temporal hierarchy of state durations across the entire cortex. Second, that there is overlap between these neural state and event boundaries everywhere in the cortex, but that this overlap should be strongest in these higher level cortical areas. Third, that neural state boundaries are shared between pairs of brain regions. And fourth, that those boundaries that are shared, so the ones that are propagating upward in the hierarchy, are also the ones that are more likely to be experienced as an event boundary. So we tested this with data from the Cambridge Center for Aging and Neuroscience. So we had 265 participants. They all watched this short movie by Alfred Hitchcock called Bang, You're Dead. And we applied a data-driven algorithm that we actually developed specifically for this purpose, for identifying these boundaries between neural states. It's called greedy state boundary search. And we applied this algorithm within small searchlight that we moved across the cortex. So here are the first results. So this is showing you the temporal hierarchy of the state durations across the cortex. So everything that's shown here in blue, purple are actually the regions showing very short-lasting neural states. The regions in red have long-lasting neural states. So the shortest neural states are found in visual cortex, auditory cortex, primary somatosensory cortex. And if we look at where the long-lasting neural states, those are actually in lateral prefrontal cortex, medial prefrontal cortex, and anterior insula. And this pattern is very reliable across participants. So here we have the data separated into 2 independent groups, and we see the exact same pattern again in this independent group. And you can see this pattern already when you just look at the raw data. So again, this is data averaged within these groups, and the top row is the visual cortex, the bottom one is the medial prefrontal cortex. And this is again this time-by-time correlation matrix. And what you can see is this very fast transitions in visual cortex and much longer lasting states in medial prefrontal cortex. So the second hypothesis was that these neural state boundaries should co-occur with subjective event boundaries. So we had a different group of participants, actually data collected by Aya Benyakov, who rated when there was an experience boundary between 2 distinct events. And we looked at how that overlapped with the neural state boundaries that we identified in a data-driven way. And what we found is that there was significant overlap everywhere in the cortex. So all the colored areas show significant overlap, but the overlap was most strongly present in those higher order regions that we would expect to show this. So again, lateral prefrontal cortex, medial prefrontal cortex, and anterior insula. So third, we tested the co-occurrence of neural state boundaries across brain regions. So for each pair of searchlights that we had in our data, we looked at how do these neural state boundaries now overlap between these different pairs. And here I'm showing you a searchlight by searchlight matrix. So here we have all the searchlights in the brain on the X and the Y axis, and all of the pairs of searchlights that are colored actually show very significant overlap across brain regions. And the ones that are white do not show significant overlap. So we found overlap between these boundaries for nearly all pairs. And we can group these regions now by how much overlap they show. And then we can identify these groups of areas that really quite closely resemble the functional networks that we're very familiar with from, for example, resting state fMRI. And finally, we looked at whether those neural state boundaries that actually propagate upward in the hierarchy, so those are the ones that are shared across regions, would also be more likely to co-occur with event boundaries. So we tested this for all these networks that we identified. So these are connections now between networks. And we found that there was indeed the boundaries that were shared were more likely to be also perceived as event boundaries. But this was particularly true for this auditory network and the anterior default mode network. So together, all these results really very nicely line up with this framework that Balsano and colleagues came up with, so that event segmentation indeed seems to be supported by this partially nested hierarchy of neural states. So we see indeed this temporal hierarchy that we expect. We see this overlap between neural state boundaries and event boundaries. We see that all these neural state boundaries are shared between brain regions, and that the ones that are shared also tend to co-occur with event boundaries. So if you're interested in trying out our method for parcellating your data into these distinct neural states, you can try out our Python package. It's very easy to use. And I just want to acknowledge all the people in my lab. and especially Jamari Utringer and Dora Kuzukara, who work with me on this, and my collaborators, Karen Campbell, Umut Güçlü, and Marcel van Gerven. Thank you very much for your attention.
Thank you for the interesting talk. We have many questions in the chat. The first one is, movies have distinct boundaries between events or scenes. Can this cause spurious results in brain imaging data that don't really correspond to natural transitions between real-life events?
Yeah, so indeed in movies, of course, you have these shots, so there's kind of these enforced transitions between distinct things that are happening. We do have some evidence that these patterns are actually also can also be observed even if you account for those kinds of shots. So in a different project, we also modeled the look at the overlap with events independent of what could be explained by simply shot transitions, and we see similar patterns there. And we see that many of these changes in neural states actually they actually really don't all align with shots, right? Some regions show the difference in the pace of these neural states between regions definitely cannot be explained by simply the nature of the movies. Yeah.
Thank you. The next question is also related to event boundaries. This person asks, are there types of stimuli that are more likely to lead to an event boundary? I would expect that this would be the case for change in location rather than, for example, a change in faces or objects.
Yeah. So it seems to be the case that the more things that change about a situation, the more likely you are to perceive an event boundary. So if there's a change in location, and if there's also a change in characters, or if there's also a change in the goal of what the characters are doing, or all of these things add up and make it more likely for someone to experience an event boundary.
Great, thank you. And what is the evidence that processing rather than only memorizing is segmented?
So there is a lot of behavioral evidence for this, especially, for example, also in literature with reading experiments where they just look at reading speed. So, for example, what you can show there is that as people are reading and they come up towards an event boundary, then after that they slow down for a while before they are back at the same pace again. So that suggests that they need some time to come up with a new event model again after this boundary, suggesting that they are segmenting the information as they go.
Great, thanks. And the next question is asking about your algorithm. So in particular, how does the algorithm that your group uses differ from the hidden Markov model event segmentation algorithm used by other groups like the Baldessano group?
Yeah. So our method is basically much simpler than the hidden Markov model method. So what our method does is doesn't have any assumptions. What it does is just try to search iteratively for where to place boundaries such that we have a maximal similarity between each neural state average and the original neural data. And we can keep placing boundaries iteratively until we have as many boundaries as there are time points, and then post hoc we determine how many boundaries was actually the optimal fit based on looking at the similarity within the states and the dissimilarity between consecutive states.
Thank you. And we have time for one more question. Which is the cortical temporal hierarchy dependent at all on the types of movies that people are watching?
Well, so that's a good question. So the results that I'm very curious to see how this replicates. So far, we've looked at Study Forest as another movie in a separate study, and there we do find a very similar hierarchy. The work that was done previously by Chris Baldessano also showed similarities, but they weren't able to explore the whole cortex, so we can't really know for sure, but there seem to be a lot of studies that at least point in the same general temporal hierarchy. Yeah.
Great, thank you so much. Thank you. Our speaker again. So we have a talk on the schedule we don't have a presentation for. Is Rodrigo Enriquez here? Okay, we're going to move on to the next talk in this session. I'm happy to introduce Claire Grahl. For this talk, please remember to type talk 4 in the app before your questions.
Hi, everyone. Thanks so much for joining me today for this presentation on my team's work on the neural processing of a nonlinear narrative. So narratives, we can consider them as causally related events over space and time, and they represent very particularly efficient and effective modes or forms of communication, in part because of how they manipulate time. Although we have to process our world in a sequential and chronological fashion, narratives are not bound in this way. When we tell a story, we can condense time or jump forward or backward. And previous research shows that information about time and temporal context is particularly important for how we construct discrete events, how we understand the relationship between events, and also how we remember and recall events later. And so with all of this in mind, we ask the question, how does the brain process time and represent temporal context while consuming a narrative that intentionally manipulates time, or a nonlinear narrative? And so to do this, we use the movie, data associated with the movie 500 Days of Summer from the Naturalistic Neuroimaging Database. And in this movie, we get to learn about a romantic relationship over the course of 500 days. But it does not do so chronologically. Instead, it jumps forwards and backwards in time using, importantly, time cues to tell the viewer when in the narrative timeline the next day will be taking place. And so with this movie, my teammate Josie carefully annotated for such things as the jumps in time associated with the time card cues, changes in location, and more, which allowed us to look into the following questions of what regions of the brain are very sensitive to changes in time, to cued changes in time, as well as how does the brain or regions of the brain represent temporal context across a nonlinear narrative. So for that first question, what brain regions are sensitive to jumps in time, we did a GLM analysis and created regressors from each of those day jumps that are cued. But importantly, each jump in time is going to be associated with a change in location. So we also created a regressor of every time there was a change in location that occurred within a day. In other words, these are the changes in location that occurred divorced from those major cued jumps in time. And to do this, we also had nuisance regressors, some low-level confounds for luminance and audio amplitude, and for this we used AFNI. And the first thing we wanted to look at was regions that are sensitive to jumps in time just compared to baseline. And right away, we're seeing sensitivity in the posterior cingulate, so centered around that medial parietal cortex. We also see parahippocampal activity associated with these jumps in time. As well, and we also took a look just at the location changes compared to baseline, and again we see parahippocampal sensitivity to those changes in location. But to look at this as a contrast, so again we're looking at the sensitivity to jumps in time over and above those changes in location to try and account for that. We see increased activation in the posterior cingulate. We see it around the intraparietal sulcus, as well as the inferior temporal gyrus and the right side dorsolateral prefrontal cortex. And we also wanted to take a note here that the parahippocampal cortex is again more active in response to changes in location, which was a nice sanity check for us, given that long association between spatial context and the parahippocampus. So we also wanted to look at if activity varies systematically with some characteristic of the jump. So for example, each jump in the movie is associated with a certain magnitude, so there's a number of days. And we did an amplitude modulated regressor, and we're not seeing anything clear here. There is some associated activity that varies based on magnitude in the primary auditory cortex, but we're pretty sure that's associated with this auditory cue that happened. That was just a page flipping that was longer during day jumps with higher magnitude. We also looked at the directionality of the jump. So for example, jumps forward versus backwards in time. And so we took the 7 backward jumps and found a commensurate 7 forward jumps of similar size and did a contrast. And we see some parahippocampal activity associated with backward jumps in time. But again, this was only 7 time points, so we're hesitant to read into this too much. But altogether, we're seeing regional sensitivity to these day jumps over and above changes in location, and this may vary according to particular characteristics of the jump. So our next question was, how does the brain represent time across a nonlinear narrative? And to do this, we did a representational similarity analysis, which for which we created 2 hypothesized matrices for how time is represented in this particular story. So we constructed these hypothesized matrices. The first one is our presentation order matrix, which would predict that days that are closer together in presentation, so the movie order that is given to viewers, are going to be represented more similarly in regions of the brain. And then our second model, our chronological order model, which predicts that regions that are days that are closer together in chronological time, so closer together in the chronological sequence of the narrative, are going to be represented more similarly. And so to construct our neural similarity matrices, at the start of each day scene, we average together the first few TRs, specifically TRs 3 through 15. So we do not include that time card cue in our data, but we account for the varying lengths of the day scenes. And then with this average, we correlated between each pair of day scenes for each individual and each ROI, and then took the lower half of this triangle and correlated it with our hypothesized matrices to derive a distribution of representational similarity values. And we did this for all of our a priori ROIs, which we took from the neurosynth parcellation. So what we see right away in this analysis, looking at presentation order, there's no strong or robust representational similarity for the presentation order of events. However, we do see strong representational similarity for the chronological order of events, and specifically in our precuneus PCC ROI, the posterior PCC ROI, and view 1. And this is stronger when directly compared in a t-test to the presentation order model. So to put this all together, in these medial parietal regions, days that are closer together in chronological time are represented more similarly, even though those days were presented out of order. We also, similar to our GLM analysis, we wanted to take a look at space, because in this movie you're constantly revisiting the same locations. So we created a binary matrix that would predict that when you revisit a day later in the movie, that will be represented more similarly when you're revisiting that same location compared to any other location. And we see a different set of regions showing robust similarity, including the temporal parietal junction, that precuneus PCC ROI, the parahippocampus A1 and V1. So again, we're seeing this nice association between the parahippocampus and the spatial context, which was a nice sanity check for us. And then we did one last analysis to compare across these models to see if any particular hypothesized model shows a stronger fit. And for those ROIs that we're seeing strong RSA values in, and we see that for the precuneus and PCC ROI, fit to the chronological model is significantly stronger than the presentation order model and the spatial location model. The same pattern of results is seen for that posterior PCC ROI, but there is no difference between the models and RSA values for the V1 ROI. So altogether, we're seeing a host of regions that are sensitive, particularly sensitive to these jumps through time compared to changes in location that happen within a day. And so we're showing that sensitivity to information about time, and it seems to be the case that regions of the medial parietal lobe are sensitive to that information and then use that information to do some on-the-fly temporal unscrambling of narrative information to then represent this sequence of events in a chronological fashion. And this lends an encoding perspective into the larger body of work showing that there's a chronological bias in memory and recall later on, and further evidence that the medial parietal lobe is this hub, a nice hub for updating and maintaining our mental models and situation models for longer narratives. Thank you so much for your time.
Thank you for that interesting talk. I want to start out with a comment, and thank you to whoever specified this as a comment and not a question. That isn't it cool we get to watch movies and do science, and I share that sentiment. But we have a number of questions as well. So how do you think the neural representation would change if the narrative time jumps were not cued? Like, for example, there was no day indicated in the movie.
This is an interesting question we've pondered a lot, and I I'm really unsure. So there are a host of movies that we can use to test that exact thing, particularly the movie Memento, which we have some fMRI data for from another team. It's been published before, where there are keyframes that are repeated when you're jumping backward and forward in time, like just a scene that is repeated, but it's not cued, but audience members can recognize that and then piece together the story. What's interesting in general, though, in the larger literature, especially from the memory literature, is that there is a bias for chronological information. We tend to recall things as chronological, and so I would say that even when it's not cued, we're still going to see some sort of chronological bias that then is reflected in memory.
Thanks. So the next question is, could the GLM for day jumps merely reveal regions sensitive to the time cue itself?
Yes, and so we moved around our regressor and saw how robust those effects were, and it was, generally consistent. And that's why, another reason why in our RSA analysis, we wanted to not include that visual cue specifically, which, we do think that V1, or that was for the RSA, not the GLM specifically, but yes, that's something that we try to account for as much as possible.
Great. This is an interesting question. So did you try regressing out visual similarity, for example, maybe similarity in DNN layers, sensitive to sort of low-level input, to see if V1 sensitivity to chronology holds, even when accounting for similarity in low-level features?
So I'm not sure I did exactly what was said in that question, but what we did try to do was stay very aware of what we were jumping to at each day jump. So right after each day jump, we have a note of, for instance, who is on screen, what location was present, the close-up. It's like, so how big, how much space is the face taking up? So all of those features, thank you so much to Josie, my teammate who led that effort. And we saw that between the presentation order of events and the chronological order of events, there doesn't seem to be much difference or no significant difference between those two when you're looking at the distribution of items jumped to, if that makes sense. So we're trying to account for those things, but I don't believe we did not do the analysis where we're looking specifically at how that V1 effect varies.
A related question is, presumably events that are sort of close together chronologically are also semantically related. Is there any way to account for this in your analyses? That was the question we were trying to dial in on when trying to, figure out how similar things are after each day jump, like what is on screen and how semantically similar that is, and whether that would confound our results. And we haven't thought of, we're still addressing this actively. We're actually looking into it as we write up a preprint. And thus far, we've done it all post hoc. We haven't figured out a nice way to concisely put this into our model right away and see how that changes results. But again, there doesn't seem to be any significant difference between those semantic features as they correspond to the chronological order of events and the presentation order of events, which are quite correlated. So I don't have a complete answer, but we are trying to address that. Thank you. And one last question. Do you think it's necessary to show the film with days in chronological order to compare? Or do you think that would benefit your analyses?
I would love to see the effect of those results, but I'm very lucky to have had so we didn't collect this data, but I'm very lucky to have had there's a paper that did a behavioral memory paper from Zhu and Kwok in 2019 that showed participants the movie in chronological order, and they also found what's called an iconicity bias in the text comprehension and discourse comprehension literature, which corresponds to a chronological bias in memory. So while we don't have chronological order here that participants saw, we do have some evidence that the chronological bias would still hold. Also, I'd like to make note of that Memento paper again, which I believe did unscramble events. They did show it in chronological order and then non-chronological order. So that would be a very interesting data set to get a hold of to retest these exact things.
Great. Well, let's thank our speaker again. Okay, so just as an update, we have received a video from Rodrigo Enriquez, so that video will be played at the end of the session after our last talk, so please stick around if you're able. Also, for those who have just joined, I'm filling in as moderator, so not able to remove questions from previous talks. So for this next talk, please start your question with talk number 5. And with that, I'm excited to introduce Esteban Villar Rodriguez.
Okay, so I'm a PhD student coming from Jaume I University in Spain, and I'm going back to the topic of language lateralization, but from a kind of a different perspective. I'm going to focus on how language lateralization affects inhibitory control in the human brain. So one of the oldest findings in human neuroscience is that some cognitive functions are hemispherically specialized. Back in the 19th century, Paul Broca described for the first time that language was lateralized in the left hemisphere. However, modern neuroimaging techniques have allowed us to be a bit more precise. When we have explored the inferior frontal cortex or Broca's area activation using an fMRI, we have seen that this holds true for approximately 94–95% of right-handed population. And when we have looked at left-handed population, this number goes down significantly. Like approximately 3 out of every 4 left-handed have this typical left lateralization, but there are some atypical cases when this is not true. We call this typical language lateralization, and it mainly it's composed of persons that have language right lateralized. Okay, they have language in the right hemisphere, or even some cases where language is like bilaterally controlled from both hemispheres. However, language is not the only function that has been described to be hemispherically specialized. We have some other examples like prosody, praxis, visuospatial attention, face processing, and word processing if you want to take it away from language. And this raises the question of what happens with these functions when language is not lateralized in the left hemisphere. Well, current data mostly support what we call the causal hypothesis of lateralization, which postulates that the lateralization of cognitive functions are like intertwined at some point of our development. So a shift in lateralization of language most of the time is followed by a shift in the lateralization of the rest of the functions. So, for example, I must remark that this isn't always happening, but it happens to most of the participants. But if language is on the right hemisphere, we would see that prosody shifts to the left, visuospatial attention shifts to the left, etc. But surprisingly, even though we usually define language lateralization by looking at the inferior frontal gyrus, no study to date has asked what happens to the function of the right inferior frontal gyrus when language is not found lateralized on the left hemisphere. So, for you all wondering that you don't know what exactly the right inferior frontal cortex does, in typical population it mediates inhibitory control. So, inhibition can be defined as the suppression of response tendencies following a cue, whether that cue is external or internal. There is a lot of neuroimaging evidence, especially by the studies of Aaron and Poldrack, that have pointed out that this inhibitory process is made possible by a right lateralized projection from the right inferior frontal cortex to the subthalamic nuclei, with additional participation of the presupplementary motor area and the right parietal cortex. So we wondered what would happen with this right lateralized network if we tried to find it in persons whose language is not on the left hemisphere. So in order to do that, we used fMRI. We modeled inhibition using fMRI by the stop signal task, as I'll just explain it really fast. It's an event-related paradigm consisting of 2 events. We have the go trials, where we present the participants with a letter string. This letter string is a word or a pseudo word, okay? A word with no actual meaning. And they just have to respond as fast and accurately as possible if it's a word or a pseudo word. But in some trials, like 20% of trials, they will be presented with a stop trial. It's mostly the same, but after being presented with the letter string, they will also hear a sound, like a beep, okay? And when they hear this beep, they have been instructed to not press any button in that trial, okay? They have to suppress their response. And this is our condition of interest for modeling this right lateralized activation of the inhibitory control network. So we had our task. Then we have our participants. We pre-selected based on a verb generation task 86 left-handed individuals. 50 of them were typically lateralized. They had a left lateralization of language. And 36 of them presented an atypical lateralization. They were 20 presented strongly right lateralization and 16 an ambilateral lateralization. So they were presented with this stop signal task, and then we calculated what has been called functional asymmetry maps. This is a really simple calculation, but it's really informative. So we have the original contrast image of the stop signal task, and what we do then is we create a copy and we flip the image by midline. So now the right hemisphere voxels are in the left, and the left voxels are represented in the right. Then if we just calculate the difference between these two images, what we have is that now every voxel is informative about how different was the bold signal between a voxel and its homotopic voxel in the other hemisphere, okay? So we calculated these functional asymmetry maps, and we compare them between typical and atypical groups. Here you have the whole brain results. Okay, these areas here are the ones that showed significant difference in functional asymmetry. So they were differently lateralized across both groups. Namely, we found inferior frontal cortex, supplementary motor area, caudate, thalamus extending into the subthalamic nucleus, angular gyrus, and middle temporal gyrus. If you recall what I just said to you earlier, we have this right inhibitory control network by Aronald Poldrack. Similarity, as you can see, it's pretty impressive with our asymmetry results that we found. But, okay, we found asymmetry difference, but in which direction? So here we have this graph for answering that question. Okay, so here we have the brain activation for both hemispheres and the 6 significant clusters that we found. Blue bars represent the typical group, and orange bars represent the atypical group. And from 0 towards the left, we have the bold signal on the left hemisphere. And from 0 towards the right, we have the bold signal on the right hemisphere. So as you can see, if we take a look at the blue bars, the typical group, the left lateralized for language group, we can see that inhibitory control has expected its tilt towards the right hemisphere. However, if we take a look at the orange or atypical bars, we can see an opposite pattern. In all 6 regions, activation is greater on the left than on the right hemisphere. So following this, we also wondered, is this effect like continuous? We can find a correlation. So what we did is we calculated the laterality index for both the stop inhibition task and the verb generation task we used to classify our participants. We found it significantly correlate, in an inverse correlation. We calculated this on the inferior frontal cortex, like defined as percularis and triangularis. It was it's not huge, but I think it's a significantly high correlation. So the more right language activation was, the more left inhibition activation was in the inferior frontal cortex. We also wondered if there existed any performance differences between the typical and atypical group, either by language or inhibition, but we did not find any difference. They were as fast to inhibit the response. They were as fast to just answer word or pseudo word in the Go trials, so we didn't have any difference here. So to sum up a bit our conclusions, we have found that inhibitory control shifts its lateralization in concordance with language. So language on the right hemisphere seems to be associated to a shift of inhibitory control to the left hemisphere. We found that this mirrored organization in atypical individuals affected not only cortical regions, but also subcortical nuclei. And we couldn't find a relation to performance of this typical or atypical lateralization. But, however, I would like to present to you something that we are currently working on following the results. If we take a look back at the correlation chart between both language and inhibition, we can see that we can classify our participants into two different groups. We have the green squares. We call these participants the segregated group because they have a strong lateralization in both language and inhibition, independently on what hemisphere each function goes, but they are really segregated functions. And then we also have the red squares, we call them integrated, because they somewhat integrate information from both hemispheres for at least one of these two functions, okay? So we use this classification for studying the lateralization of the inferior frontal cortex, and we found some really interesting results. For example, we found that integrated individuals, the ones that have weaker lateralization in at least one function, were slower when inhibiting their responses during the stop signal task, and also made more mistakes during a reading task. Also, we found that integrated individuals had a stronger interhemispheric connectivity, both functionally and structurally. This result is really interesting because it's in line with what has been recently described in this kind of atypical population. So we are really eager to wrapping up these results and present them to you more officially. And that was it. I would like to thank my colleagues at Universitat Jaume I back in Spain, my research group, the NNF, the Spanish Ministry of Economy for making this research possible, and of course, thank you all for your attention.
Thank you. The first question is, are all of your participants monolingual? And what language or languages do they speak?
No, in fact, they were bilingual. We are from a part of Spain, near Valencia, and everyone there talks Spanish and Valencian, so they were all bilinguals.
Thank you. And how did you account for anatomical asymmetry in your brain fluid?
Yeah, that's a really good question. When preprocessing the fMRI data, we use a special tissue probability map that is symmetrical.
Thank you. And what threshold did you use to determine language laterality in your analyses?
What? Could you repeat, please?
What threshold did you use to determine language laterality?
What?
Threshold?
Okay. We used a laterality index lower than 40. Yeah, it's not the most used threshold, but we were really interested in this segregated versus integrated analysis we did later. So we wanted to have a seizable sample of integrated individuals.
Thank you. This person noted that atypical participants seem to have a dominant hemisphere less activated than typical participants.
What?
Is it comparable?
Could you repeat, please?
Yeah, so atypical participants seem to have a dominant hemisphere less activated than typical participants. Is it comparable?
No, I don't think I understand. Could I read the question?
Does somebody want to clarify? If you ask the question, do you want to step up to the microphone and clarify?
I think if I just read it, can I go there?
Sure. Can you hear me? Here, I can show you.
Actually, we did not find that in our data. I mean, there were some clusters in these 6 regions that had basal levels in bulb activity independently of the hemisphere, but we did not find differences in in the strong of lateralization between both functions and the direction on the hemisphere.
Okay, great. Well, thank you so much for this great talk, and thank you to all of our speakers and you for joining us. Unfortunately, we don't have a video to show last, so this is the end of our session. Enjoy lunch and the rest of the conference.